In this lesson, we're going to be focusing on eukaryotic cell structure. So with our last lesson, we focused on prokaryotic cells, which we pretty much just exclusively focused on bacteria. In this lesson, we'll be talking about eukaryotic cells, which may sound familiar because most of what you've probably studied in biology is based on eukaryotic cells, like human cells, plant cells, other animal cells. We'll be focusing on microorganisms that have eukaryotic cells like protozoa and fungi, but there are some similarities between different eukaryotic cell types. So these are all the different structures that we're going to be talking about. In this column, we have the structures that all eukaryotic cells have. So regardless of whether we're talking about fungi or humans or plants, they would all have these kind of essential core eukaryotic structures. And then over here, we have some of the structures that specific eukaryotic cell types would have, but not all. So for example, plants have chloroplast. Things like fungi have cell walls, whereas humans wouldn't, that type of stuff. So when we look at a eukaryotic cell versus a prokaryotic cell or a bacterial cell, one thing I do want you to acknowledge is how much more complex eukaryotic cells are. So eukaryotic cells, they're typically much larger, and they do have all these membrane-bound organelles, which you're probably quite familiar with. Things like a nucleus, and endoplasmic reticulum, and Golgi apparatus, and mitochondria, and lysosomes, and vacuoles, all different types of stuff. And then over here, when we look at a bacterial cell, which is a prokaryotic cell, they do not have any membrane-bound organelles. They have their genetic material here that's within the cytoplasm, but it's not encased in any type of membrane, so it's not a nucleus. And they have some ribosomes, but internally, they're much less complex than what we would see with a eukaryotic cell type. So we're going to start out this lesson similar to how we started out the last lesson with prokaryotic cells, in the sense that we're going to start out on the exterior of the cell and then kind of work our way in. So we're going to start off by talking about various external structures that eukaryotic cells can have. One of the very common external structures that eukaryotic cells can have are flagella. Just like in the last lesson, flagella still serve the role of the motor appendage, so they are what allows the cell to be mobile. Typically, they respond to various chemical signals. We know that as chemotaxis, and if they move in response to a chemical signal, if they move towards something in response to a chemical signal, we call that positive chemotaxis. And then if they move away from something potentially harmful, we would refer to that as negative chemotaxis. So functionally, both prokaryotic and eukaryotic cells use flagella for movement. But structurally, there are some important differences between prokaryotic and eukaryotic flagella. And those differences are summarized here in this chart. So if we're looking at a bacterial cell or a prokaryotic cell, their flagella are much smaller and much less complex. A prokaryotic flagella is typically just one single filament. So this is what we're looking at here. It's just one single filament. With eukaryotic flagella, they're typically much thicker and much more complex, which is what we're seeing right here with eukaryotic flagella. Eukaryotic flagella are actually made up of 11 filaments called microtubules. There would be 2 in the center, and then 9 that surround those 2 in the center. That's where we get the 9 plus 2 microtubule arrangement. But you can see that it makes for much thicker, much more complex flagella as compared to a prokaryotic flagella, which is just one single filament. And then quantity is different. So with prokaryotic cells, they can just have one flagella, and that would be considered a monotrichous flagella arrangement. But more often than not, they have numerous flagella, hundreds, potentially even thousands of flagella. So they typically have a higher quantity of flagella as compared to eukaryotic cells. Eukaryotic cells typically just have 1, maybe 2, but no more than that as far as the number of flagella, which makes sense because with prokaryotic cells, their flagella are much thinner, so they need more of them in order to get a good range of motion, whereas with eukaryotic cells, their flagella are much thicker, much more complex, so they don't need as many of them to still get that same degree of movement. And then the last difference is the actual type of movement. So with prokaryotic cells or bacterial cells, when their flagella move, they move in a circular motion. So when they want to run, they turn counterclockwise. And then when they want to tumble, they turn clockwise, but it's a circular motion. With eukaryotic flagella, they move in more of a whip-like motion versus a circular motion. So the actual type of movement is different. An additional motor appendage that only eukaryotic cells can have is called cilia. Now, cilia are exclusive to eukaryotic cells. They're used for movement, but only eukaryotic cells can have this structure. Prokaryotic cells do not have cilia. In a prokaryotic cell, the only motor appendage is the flagella, whereas in a eukaryotic cell, they could have flagella, but they could also have cilia. Or there's even opportunities for them to have both. So with cilia, they're very similar to flagella as far as composition. They're made up of that same 9+2 microtubule arrangement. So a cilia is going to be made up of 11 microtubules. They are much shorter than what we would see for flagella on a eukaryotic cell, and they're much more complex. So for example, this is a protozoan cell, and all these little short hair-like fibers all over this cell are cilia. They're much shorter, but there can be a higher quantity of them. But what those cilia are going to do is move in a back and forth type motion, like a boat roving through water, to propel the eukaryotic cell through an aqueous environment. So this is a very common motor appendage for protozoan to have, which are commonly found in water. They will just move those cilia back and forth in order to move through the water. And cilia are also found in cells within our body. We have cilia throughout the respiratory tract and the female reproductive system to help with movement of various things as well. But cilia are exclusive to eukaryotic cells only. As far as the glycalyx, we covered the glycalyx with glycalyc structure in the prokaryotic lesson. We said that a glycalyx could be present either as a slime layer that loosely surrounds the bacterium and aids in dehydration, or it could be present as a capsule, which is tightly bound to the bacterium and protects the bacterium from phagocytic cells. Those same concepts are consistent here in the eukaryotic lesson. So eukaryotic cells can still have a glycalyx, and that glycalyx can be either a slime layer or the capsule. So all that information is the same between the two. The cell wall, there are some similarities between the cell walls of prokaryotic and eukaryotic cells, but there are some core differences. So both prokaryotic and eukaryotic cells can have a cell wall, and that cell wall serves the function of maintaining shape and support for the cell. Not all eukaryotic cells have a cell wall. For example, humans don't have a cell wall, but something like fungi would. With eukaryotic cells that have a cell wall, it's really important to remember that the cell wall is made up of something different than what bacterial cell walls are made up of. So bacterial cell walls are made up of peptidoglycan. With eukaryotic cells, if they have a cell wall, it is not composed of peptidoglycan. For example, fungi are eukaryotic. They have a cell wall that helps them maintain shape, structure, and support, but the cell wall does not contain peptidoglycan. Instead, it contains this component called chitin. So here we're looking at a fungal cell wall, and inside that cell wall we do have chitin. If this were a prokaryotic cell wall, it would look similar, except the cell wall would contain peptidoglycan. But as far as functionality, they have the same function, and that's to maintain cellular shape and support. The cell membrane is very similar between the two different types of cells, being prokaryotic and eukaryotic cells. So as far as the cell membrane or the cytoplasmic membrane, even in eukaryotic cells, it's composed of phospholipids. It's composed of a double layer of phospholipids, which we can see here. There's 2 layers of these phospholipids. Embedded in the layer of phospholipids, there are proteins, these big blue things here, that are used to help with transport. So they are quite similar. One unique difference, though, is that only eukaryotic cell membranes have sterols embedded into their cell membrane. So this is a unique trait for only eukaryotic cell membranes. Prokaryotic cell membranes are not going to contain sterols. And the reason for that is prokaryotic cells, they have a cell wall that contains peptidoglycan to help maintain their structure and support. But if we think about human cells don't have a cell wall, but they still need something to maintain their shape and support so they don't kind of collapse in on themselves. So what our cells have is in the cell membrane, that's where we have sterols embedded. And sterols help maintain that rigidity and support, which is especially important in cells that don't have a cell wall that would typically serve that function. So eukaryotic and prokaryotic cell membranes are very similar in what they're composed of as far as phospholipids and having proteins that aid in transport, but the one distinct difference is that eukaryotic cells contain sterols in their cell membrane to help with rigidity and support. As far as function, the function's the same regardless of what type of cell we're looking at. Overall, the cell membrane's primary function is transport. It's selectively permeable. It controls what goes into the cell and what goes out of the cell as far as waste. Now, the prokaryotic cell membrane has additional functions as far as ATP production and nutrient processing, but still that primary function is going to be transport. So kind of starting to work into the cell, probably the most predominant structure inside of a eukaryotic cell is going to be the nucleus. All you really need to know for the context of this course, because you have learned this material before, is that the nucleus is used to house the genetic material, or the DNA. So inside of the nucleus we have DNA in the form of chromosomes. Only eukaryotic cells can hold their DNA inside of a membrane-bound nucleus structure. Prokaryotic cells lack a nucleus, so their DNA and their chromosome is typically just freely associated with the cytoplasm. It's not contained in any type of membrane-bound compartment. So the nucleus is something that is exclusive for eukaryotic cells only.